In this lecture, we're going to look at muscle tissue. Now, muscle tissue does one of two things. It either contracts or it relaxes. It does not push. In other words, if I want to bend my arm at the elbow, then my bicep muscle has to contract. Now, in order to straighten my elbow, well, my bicep can't push my arm back out. It has to relax. And then another group of muscles, an antagonistic group of muscles, is going to have to contract, and that would be my triceps. And they would contract and straighten my arm back out. So remember, muscles either contract or relax. And they're going to take chemical energy and they're going to change it into mechanical energy. Now, muscles tissue comes in three types. We have skeletal muscle, and this attaches to bone, skin, or fascia. We say it's striated because there are these light and dark bands which are visible under the microscope. And it's under voluntary control of contraction and relaxation, and this is what it looks like. Now, again, we consider skeletal muscle striated and voluntary. Striated because of the stripes, and that's because of the way we have these sliding filaments that slide together to create this banding pattern, where we have thick filaments and thin filaments, and as they overlap, that creates the stripes. And we say it's voluntary. So again, skeletal muscle is striated and voluntary. We have conscious control over it. The next type is going to be cardiac muscle. Cardiac muscle, of course, we find in the heart. It's also striated in appearance because of the way it contracts. But instead of being under voluntary control, it's under involuntary control. We don't have conscious control over it. And we say it's autorhythmic because of built-in pacemaker. Now, if we take cardiac muscle tissue and put it in a petri dish of nutrients in a tissue culture, and separate the fibers out, each of these little muscle fibers are going to contract on their own. When they come together, they tend to contract in unison. So again, this is why we say they're autorhythmic, because of the built-in pacemaker within these muscle cells. And when the heart's put together as a whole, of course, there's a whole conduction system to coordinate that beating. Now, if these muscles tend to start contracting on their own, and we get just this asynchronous quivering effect, we say that muscle tissue is in fibrillation. Okay, fibrillation. And it's kind of described as like a bag of worms appearance, where it's just kind of twitching and quivering. And how do we correct fibrillation is with a defibrillator. And again, that's where you put the paddles on the chest, send electricity from one paddle to the other. That causes the heart muscle to contract all at once. And once the electricity stops, then all of those muscle cells relax at once, and hopefully they're back in sync once again with their contraction. And this is what it looks like. Again, it's going to be kind of this teased appearance. The nuclei are centrally located, as opposed to the skeletal muscle, which is more cylindrical and has the nuclei off on the periphery. And actually, each of the muscle cells are multinucleated, whereas the cardiac muscle, the nucleus is centrally located. Okay, the next one is going to be smooth muscle. And we find it in a lot of different places. If you remember when we talked about the skin, it's attached to hair follicles in the skin. Remember the erector pili muscle? That's an example of a smooth muscle. It's also found in the walls of hollow organs, such as blood vessels or your gastrointestinal tract, your intestines, basically. It's smooth because it is non-striated in appearance. Okay. And it's also involuntary. We have no conscious control over it. But here's what it looks like. And so, just to recap, skeletal muscle is striated and voluntary. We have conscious control. Cardiac muscle is striated and involuntary. And smooth muscle is non-striated and involuntary. We don't have conscious control over it. So let's look at the function of muscles. It helps to produce body movements, helps to stabilize body positions. It's also going to regulate organ volumes, and one way it does that is with these bands of muscle called sphincters that close off each end of an organ. Let's talk about the stomach, for instance. The stomach is basically a muscular bag, and as food goes in, first of all, it goes through the lower esophageal sphincter, and then through the cardiac sphincter into the stomach. At the opposite end of the cardiac sphincter, we have the pyloric sphincter. Now, typically, when food goes into the stomach, both the cardiac sphincter, the lower esophageal sphincter, and the pyloric sphincter are closed, and you get this kind of churning action that takes place. And as the foodstuffs get broken down into something we call chyme, the pyloric sphincter will open a little bit and let a little piece of that chyme go off into the small intestine. Okay, so that's one example. Another place where you see sphincters are sometimes in capillaries. We have these precapillary sphincters that will allow a certain amount of blood flow to and from organs. Another organ where we see regulation of volumes would be the heart. The heart's basically a four-chambered hollow muscular sac, basically. And when the heart relaxes, the volume increases within those chambers, and so blood can fill up in those chambers. When the heart contracts, the volume becomes less, the pressure increases within the heart, and it ejects the blood out. By the way, that's Boyle's law. Boyle's law is where if there's an increase in volume, there's a decrease in pressure. If there's a decrease in volume, you get an increase in pressure. Volume and pressure are inversely proportionate. So that's going to be important later on when you start looking at, the heart and the lungs and what have you. Okay, so, and then we have movement of substances within the body by muscle, such as the movement of blood or lymph, urine, air, food, fluids, sperm, and there's a group of words that shouldn't be in one sentence. So let's move on. And then producing heat. We have involuntary contractions of the muscles or shivering is going to increase metabolism, and a byproduct of metabolism is heat. But just the fact that you're moving, forget about shivering, but just movement itself is going to, again, expend a little bit of heat energy because, again, like I said, anytime you have increased metabolism, a byproduct of that is going to be heat. Okay, looking at properties of muscles, we have excitability, and muscles can respond to chemicals that release from nerve cells. There's conductivity, and that's the ability to propagate electrical signals over the membrane. This is why we're able to measure those signals on the heart, and we can get what we call an EKG or an ECG, electrocardiogram. Okay, but we can also do that with our skeletal muscles too, with an EMG, electromyography. Okay. Also, properties of muscle, we have contractibility, and that's the ability to shorten and generate a force. Then there's extensibility, and that's the ability to be stretched without damaging the tissue. But of course, that's within normal physiological ranges. You exceed that normal physiological range, you will damage the tissue. And then elasticity, and that's the ability to return to its original shape after being stretched. Once again, you can't exceed that normal physiological limit. If you do, then it's not going to return to its normal shape, and you can even damage or break the tissue. I mean, think about elastic waistband and pants. You know, if you overstretch it, you know, it may not come back to its regular size, right? It stays overstretched. Same thing happens with muscle tissue. But if you put a nice, smooth, steady, gentle stretch into it, you can put a nice permanent elongation into that muscle. If you do it fast, you can break the muscle. Think about silly putty. I don't know if anybody out there has ever played with silly putty, but in my practice, I used to keep a little egg of silly putty to show my patients, especially those who had been in a car accident. Because if I take that silly putty and I pull it slowly, what happens to it? It stretches out, right? But if I take that same silly putty and pull it fast, what does it do? It snaps. And again, that's kind of like the muscles. You can put a nice stretch in your muscles, but like, for instance, in a car accident where you get the whiplash, you all of a sudden put a fast stretch into that muscle, and that's when tissue is damaged. Okay, looking at skeletal muscle connective tissue. We have what's called superficial fascia, and that's going to be the loose connective tissue and fat underlying the skin. Again, another name for that is the hypodermis or the subcutaneous tissue. All three of those are the same name. It's just that when we're talking, you know, as an anatomist, we're calling this the superficial fascia. Then we have the deep fascia, and that's dense, irregular connective tissue surrounding muscle. If you've ever eaten a steak and you know the gristle, or what they call the silver skin, that's this white band going around the steak, you know, very inedible. You have to cut that off. You can't chew that. Okay, that is what we're talking about here. That's the deep fascia. Again, it's made of that dense, irregular connective tissue. And again, it wraps around muscle groups. And then connective tissue components of the muscle include the epimysium. Think about what's above your dermis, epidermis, right? Well, what surrounds the whole muscle on the outside? Well, it's the epimysium. Myo means muscle, and so that's where that term mysium comes from, refers to muscle. So epimysium is above the muscle, basically. So that surrounds the outside. Then we have these bundles of muscle fibers that are called fascicles. And by the way, when I say muscle fibers, that's synonymous with muscle cells. So muscle cells, muscle fibers means the same thing. Okay? And so these fascicles are made up of bundles of 10 to 100 muscle cells or muscle fibers. And what surrounds them is the perimysium. Peri means around. Think of perimeter. Perimeter is like around the outside. Okay, the perimysium is surrounding the outside of these bundles. You know what this reminds me of? When I was growing up, um, in the fields surrounding where we live, they must have been putting in phone cable, underground phone cable. And we would find these pieces of phone cable. They must have been probably a couple inches in diameter. They were thick, okay, pretty heavy. But inside was all this really colorful phone wire, which was, fun to make things with. I used to do all kinds of stuff with that. But on the outside of this cable was this thick black plastic. Well, that's kind of like the epimysium. Okay. Well, back in my day, even as a kid, I mean, it was no big deal. We all had our pocket knives with us and everything. And so I'd pull out the pocket knife and start carefully cutting away that thick black plastic. And we would find these bundles of wires. Well, that would be like the fascicle. And there was this kind of a white tape that was wrapped around these bundles of wires. Well, that would be like the perimysium. Okay. Well, on the inside of the muscle, if we get to the innermost of these fascicles, well, it's made up of the individual muscle cells or muscle fibers. And what's the term we use for inside? And that would be endo, right? So the endomysium. So this would be like when I would unwrap that tape from that bundle and I would just have the individual wires. Think about those as the individual muscle cells. Well, what surrounds wire? Well, you have that plastic insulation, right? That's what covers the wires. That would be like the endomysium. Okay. And so going from the outside the outside, we have the epimysium. Then we have surrounding the bundles, the perimysium. And on surrounding the individual muscle cells themselves, the endomysium. And I've got a picture of that. I'll show you in just a moment. But actually, here, let me skip over to the picture. We'll come back and finish that slide in just a second. So here's a tendon. Here's that deep fascia that we talked about. Surrounding the whole muscle itself, again, is going to be the epimysium. Okay, so the epimysium. Then we see these bundles. Okay, these bundles are the fascicles. Let's pull one of these fascicles out. Okay, so here's a fascicle. Now notice the connective tissue going around the fascicle. Okay, that is the perimysium. Perimysium. Okay. And we pull that out and we notice the individual muscle cells or muscle fibers. And that's about as far as inside you can get when you're looking at these connective tissue components. What's the other term for inside? Endo. So this is the endomysium. Let's suppose we are going to make a model of this. And maybe I took, those barbecue skewers, like the little bamboo skewer sticks. Maybe I'm going to use those as representing my muscle cells. Okay. And maybe I take some saran wrap and I wrap it around the muscle cell and I extend that saran wrap past the sticks. Okay. Well, I do up a whole ton of these, and then maybe I take about 10 of them and I pull it into a bundle. Now, what's a bundle of those muscle fibers called? A fascicle. So I've got a fascicle of these skewers. So I wrap them up with, 10 of those. I wrap those up with saran wrap, but again, I extend the saran wrap past, the sticks. All right, so that saran wrap that I wrap the individual sticks with is the endomysium. The saran wrap that I'm wrapping up the fascicle with is the perimysium. Okay, now we've made a whole bunch of these fascicles. I put all of those together and I wrap them up in some more of the saran wrap, and I extend it past, again, the bundles. Okay, and so that would be like my epimysium. So what about all that extra saran wrap that's hanging off each side of this muscle model I just made? Well, that is going to be our tendon. Okay, right here, the tendon. So all the connective tissue layers that extend beyond the muscle belly form the tendon. And this is why tendons are so strong. Can you break a tendon? Sure you can. You can break anything. But it's very difficult to break a tendon. You can't just pull it off. It's not like glued onto the end of the muscle. So it's not just going to pop off. Okay? It is actually integrated into the entire muscle, all the way down to the cells, to the fascicles, to the whole bundle of muscle itself. So this is what makes it so strong. Okay. Oftentimes too, especially if the muscle is nice and healthy and everything, and if you're still pretty young. Now with me, as you age, the tendons can become calcified and they can snap. And I've seen that happen with guys getting up in their 30s, 40s, still playing basketball, and they go to make up a layup shot and they jump, they hear a snap, and they come crumbling down into a little heap below the net. Well, their Achilles tendon has become kind of calcified and snapped. Okay. But in a young, healthy person, oftentimes, instead of the tendon actually snapping, it'll actually rip right off the bone and take some of the bone with it. We call that an avulsion fracture. Okay. And yeah, again, that's how tough, tendons typically are. Looking at the nerve and blood supply of muscles. Now, each muscle is supplied by a nerve, artery, and two veins. We call that a neurovascular bundle. And each motor neuron is supplied by multiple muscle cells at what we call the neuromuscular junction. And each cell is supplied by one motor neuron terminal branch, and it's in contact with one or two capillaries. And nerve fibers and capillaries are found in the endomysium in between individual cells. Now, I mentioned that skeletal muscle cells are multinucleated. Well, how does that happen? Well, that's because of the way they're made. As you can see, we have myoblasts. Remember, blasts make things. They're going to fuse to form a mature skeletal muscle fiber or cell. And so every mature muscle cell develops from 100 myoblasts that fuse together in the fetus. This is why it's multinucleated. The mature muscle cell now cannot divide. This is important. You think when you go to the gym and you start working out that you're going to get more muscle cells. You don't. As a matter of fact, any skeletal muscle cells that get injured or die off typically will not be replaced. Okay. Now, sometimes some new cells could be regenerated. but usually not. So you're saying then, how come the muscle gets bigger then if we're not going to make more muscle cells when you work out? Well, muscle growth is a result of cellular enlargement, not cell division. So, in other words, it's hypertrophy or enlargement of those individual muscle cells themselves. You don't get more cells, the cells just get larger. See, inside the cells are these filaments that when they contract, that's what makes the muscle contract. Or when they slide together, that's what makes the muscle contract. That's what I'm trying to say. And so as you work out and you start to exceed the limit of that muscle, well, it does what it can to keep up with, being able to lift that certain weight or what have you. And how is it going to do that? By adding more of those sliding filaments and mitochondria and what have you inside that cell. And it's like stuffing more stuff into a sack. That sack's going to expand and get bigger. And that's what we're doing here. We're just stuffing more of those sliding filaments and mitochondria, etc., into that cell. It gets larger. Now, satellite cells retain the ability to regenerate new cells, but typically that is not enough to keep up with any type of muscle damage. So you are not going to typically get more skeletal muscle cells if they get damaged or destroyed. And so let's take a look at a muscle fiber or muscle cell, and also the myofibers. Now, again, this is a muscle cell. Doesn't look like your typical cell that we've looked at in the past. It's going to be cylindrical. You notice it's multinucleated. Okay. And again, we call it a muscle fiber as well because of it being kind of long and stringy. Now, it's going to have a lot of the same parts that we find in regular muscles, except we're going to give them typically a different name. Remember the cell membrane on a regular cell? I said that another name we could call that is the plasmalemma. Well, with muscle, instead of calling it a plasmalemma, we're going to call it a sarcolemma. Okay, so the sarco means flesh. Okay. And that's the term we use for muscle. Like a sarcoma is a tumor of muscle. Okay. Sarcolemma is the plasmalemma or cell membrane of the muscle cell. So let's think about that for a second. What do you think we call endoplasmic reticulum inside of a muscle? Well, it would be sarcoplasmic reticulum. What do we call cytoplasm inside of a muscle cell? It's sarcoplasm. Okay. What do we call the mitochondria inside of a muscle cell? Trick question. It's still mitochondria. Sorry about that. Okay, moving on. The sarcoplasm, again, that's the cytoplasm of the muscle cell, is going to be filled with tiny threads called myofibrils and myoglobin. Now, myoglobin is the red-colored oxygen-binding protein. It's very similar to hemoglobin. Hemoglobin is found inside your red blood cells, and that's what carries oxygen to the body. In fact, anemia is when you're not making enough hemoglobin, and so that kind of lowers your oxygen-carrying capacity. When oxygen binds to, let's just talk about hemoglobin for a second inside red blood cells. When oxygen binds to that hemoglobin, we get kind of a chemical reaction. Okay, and we say that hemoglobin is reduced, and what happens is it becomes brighter red, okay, in color. That's why if you draw blood from an artery and the person is oxygenated enough, that blood is going to be bright red. Okay? Now, typically, we're used to getting blood drawn from a vein. Venous blood basically has given up its oxygen to the tissues. So when we look at venous blood, it's typically a very dark red. In fact, when you look at veins through your skin, they look blue. Okay, they're not blue, it's actually dark red, but just combining the blue with the flesh color here, your skin tones, gives it that bluish appearance. Okay, well, same thing with myoglobin. The more oxygen that binds to it, the more red looking that particular tissue is. This is why we say meat, like beef, is red meat. Okay, that red comes from the myoglobin. By the way, I don't know if you realize that, but when you eat steak or chicken, okay, that is muscle that you're eating. Fish, that's the fish muscles that you're eating. Okay? Well, there is a gas other than oxygen that likes to bind to hemoglobin, and it's pretty dangerous, actually. And now, hemoglobin, again, in the red blood cell, there are 4 heme groups within there, and so there's 4 spots for oxygen to bind to. If they're bound to all 4 spots, then that hemoglobin is 100% saturated. When we talk about doing a saturation, where we put that little thing on your finger and check for a saturation, okay, that's what we're talking about. How saturated is that hemoglobin? Okay. And so what happens with this gas, and have you guessed what that gas is yet? It's carbon monoxide. Okay, carbon monoxide. Carbon monoxide will bind to all of those heme groups, totally saturating the hemoglobin. And now oxygen can't bind to it. And the thing is, it doesn't let go. See, when oxygen binds to hemoglobin, it's a very, what should I say, loose binding to that hemoglobin, because when the blood goes to tissue that needs oxygen, it has to be able to release that oxygen so that the tissues can take it up. If it hangs on tight to the oxygen, it's not going to let it go. And that's what happens with carbon monoxide. That's why it's so dangerous, because it binds and doesn't let go, and now oxygen cannot bind to it. So let's guess what color that blood is that's totally saturated with carbon monoxide. Do you think it's going to be bright red? Okay, or do you think it's going to be dark red? Okay, you might have guessed dark red because there's no oxygen bound to the hemoglobin, but that has nothing to do with the coloring. Again, it's all chemistry. The fact that the hemoglobin is saturated, or what we call reduced, and the fact that typically when we look at oxygen bound to hemoglobin, it's not 100% bound, but carbon monoxide is 100% bound. The blood is not only bright red, it is cherry red. Super bright cherry red. Okay? And so at which point we have to get that patient into what we call a hyperbaric chamber, which pumps up oxygen, 100% oxygen under pressure to try to dislodge that carbon monoxide and get oxygen to the tissues. Okay. All right. So what does that have to do with muscle? Now going back to the myoglobin and going back to beef. Okay. The grocery stores and meat industry has discovered that, when you buy a steak and maybe the next day, it's not looking so good. I know when I buy my red meat, I want it to be red. Okay. But a couple of days later, it might start to look kind of gray and not very appetizing looking. So what they have discovered is if they pump a little bit of carbon monoxide into the packaging, that binds to the hemoglobin, or myoglobin, I should say, and really brightens up the look of that meat. So it makes it look a lot more appetizing and a lot more fresh. And that's the only concern, is that you want to check the sell-by date on it because it might look fresher than it really is. Okay. But other than that, it's not going to cause a problem. So it's not dangerous because as you're cooking it, it gets released anyway. Just don't breathe the contents of the package, as you're opening it. Anyway, so myoglobin's going to react just like hemoglobin is. And so myoglobin is going to store that oxygen so that when the muscle overexerts, it has an extra store of oxygen, not relying on just what's coming out of the blood. Okay, I know that was a little long-winded. Sorry about that. Okay, now I've got a weird question. Has anybody ever been to Boston? Okay, well, if you've ever gone to Boston, or if you ever do plan on going to Boston, don't plan on driving in Boston. Bostonians are crazy drivers. Okay, and if anybody is from Boston who's listening to my videotapes, no offense, I love Boston. Boston's cool. Okay, I love visiting. Okay, if you do visit, you have to check out Legal Seafood. And if you're from Boston, you've probably been to Legal Seafood. Awesome clam chowder. Okay? So you don't want to drive around Boston. So how do you get around? Well, you take their subway, and they call their subway the T. Okay? So the T. And you can get all over. I've gone there. I love visiting the old ship, the Constitution, and for my students, you probably didn't realize I went to Harvard, right? Did you know that? Yes, I went to Harvard. I went there and I checked out the bookstore and bought a nice Harvard hat and also a little briefcase. Walked around the grounds. No, I didn't study at Harvard. I just went there one day. So it was a lot of fun. Anyway, so you take the T. And how do you get down to a subway? Okay, you take stairs, and that goes down below the earth, and you're down in the T, and you can catch the train. So what does that have to do with anything? Well, when we're looking at action potentials are these electrical impulses that are going to go across the muscle sarcolemma, basically, and they have to find a way to get down inside the muscle cell to do their thing. Okay? Well, again, just like driving in Boston, you don't want to drive there. You want to take the T. And so same thing with the sarcolemma. You're up here, and then you go down into the T tubule or transverse tubule. And again, these T tubules are invaginations of sarcolemma into the center of the cell. And it's going to be filled with extracellular fluid. And that's just going to help conduct that electrical impulse, or that action potential, as we call it, down into the cell. And that's what I just said. So it carries muscle action potential down into the cell. Now, we're going to find a lot of mitochondria which lie in rows throughout the cell because, again, muscle is very active. Mitochondria, if you remember, is your ATP factory, and ATP is the energy currency that the cells use. And so these mitochondria are near muscle proteins that use ATP during contraction. Okay. Now, looking at myofibrils and myofilaments, muscle fibers are filled with threads called myofibrils separated by sarcoplasmic reticulum. And all this yellow here is the sarcoplasmic reticulum. And again, we can see in the blue here, these are the T tubules. And so the myofilaments that we find in the myofibril here are thick and thin filaments, and they're contractile proteins of the muscle. So this little unit here is going to be the contractile unit made up of these thick and thin filaments. And so we're used to talking about and looking at inside of a cell the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Well, the sarcoplasmic reticulum is very similar to smooth endoplasmic reticulum in non-muscle cells. So again, it's a system of tubular sacs. The difference is in muscle, it's going to store calcium. Okay? Calcium's what is needed for the muscle to contract if calcium is released and is present. Now, in a relaxed muscle, that calcium is pumped back out and into and stored into the sarcoplasmic reticulum. And again, when this calcium is released, that's what's going to trigger a muscle contraction. And now let's look at these two words. And can you pronounce these? Go ahead and pronounce that word. Okay, that word is atrophy. Now pronounce that word. Okay, if you said hypertrophy, ooh, not so good. The way you want to pronounce this word is hypertrophy. So atrophy, not atrophy. That's what you put up on your mantle. You call it atrophy. And this is hypertrophy. So atrophy is the wasting away of muscles. And it's caused by a few different things. You can have disuse atrophy, in other words, you use it or lose it. If you've ever had a cast, maybe on an arm or a leg or something, oftentimes when that cast is removed, that muscle's kind of shriveled up looking. That's because it has atrophied. Now we can get atrophy in another way, and that's by severing a nerve. Okay, and we call that denervation atrophy. So perfectly good muscle, but we cut the cord going to it. Now it's not going to function. And the transition to connective tissue cannot be reversed. Remember all that connective tissue? The endomysium, the epimysium, the paramysium. If everything has atrophied away down to that point where it's just connective tissue, we can't reverse that. Now going back to like if the muscle has shrunk down because you've had a cast on or something, there's still some muscle tissue there, so that muscle can be rehabbed and built back up. Okay. Now, there we go, hypertrophy. This is where we have an increase in the diameter of muscle fibers. And that's going to result from very forceful, repetitive muscular activity and an increase in myofibrils, sarcoplasmic reticulum, and mitochondria. That's what I talked about before, where you're not getting more muscle cells. You're just building up the individual cells with more of these myofibrils, sarcoplasmic reticulum, and mitochondria, and causing the cell to enlarge or hypertrophy. Now looking at filaments and the sarcomere, and by the way, the sarcomere is your functional unit of the muscle cell, and it's going to go from Z disc to Z disc. Okay, so Z disc to Z disc, and I'll mention that a few times, so it must be important. Now, the thick and thin filaments overlap each other in a pattern that creates the stripes or striations. The light areas are going to be the I bands, and the dark areas are going to be the A bands. Like, for instance, from here to here would be an example of an I band. From here to here, this area would be an example of an A band. Now, the I band region contains only thin filaments, and they're arranged in compartments called sarcomeres. And again, they're separated by the Z discs. So, sarcomere, Z disc to Z disc. In the overlap regions, you have 6 thin filaments, which are going to surround each thick filament. Okay, so 6 thin filaments surround each thick filament, and that's what we see here. Now, looking at thick and thin filaments, we have the supporting proteins, which is the M line, tighten filament, and also the Z disc, and that helps to anchor the thick and thin filaments in place. The thick filaments are right here, and this is going to be made up of your myosin, and these little thin filaments here are going to be made up of what we call actin. And again, here we can see the I band. Here we see where the overlap is. This is the A band, and again, here's an I band once again. This is what it would look like under an electron microscope. And let's go ahead and take a look at a little video that kind of shows and explains all of these different things, like the Z discs, the M lines, the H zones, the I bands, and the A bands.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.